Welcome everyone on this very special lecture. And I would like to introduce our guest speaker, who is Hans Hoffman, Professor Hans Hoffman from University of Texas, from Austin. And he's basically a neurobiologist working on the cichlid fish model systems, and he's really interested in interactions between physiology, the environment, and social behavior. But today he will talk about something else. He's very keen on talk about secrets of a scientist's life, how to prepare good presentations, how to organize your research career. And today's talk will be about especially talks, presentations. So I hope that this will be very useful for you, because Hans is a very talented and good lecturer. So I hope you will enjoy. And you can start your presentation now.
Great. Thank you, Balash, for the introduction. Thank you all for coming late at 5 o'clock. Normally you want to be at home and get dinner ready or something. Okay, I'm going to talk to you about how to give a scientific presentation, but I need to know a few things about you first. One thing I'd like to know is who in this room has already given a scientific presentation in a class, project, or about their own research or so. Many, but not most of you. Okay? And have you attended other people's scientific presentations? Lectures, seminars? Yes. Probably all of you have done that. Have you all ever fallen asleep listening to someone? If you say no, I don't believe you. I fall asleep all the time, right? So, and the question is, why is that? Sometimes you fall asleep because you're tired. And, no matter what happens up on the stage, you just cannot stay awake because, you've worked for too long, or maybe you were at a party for too long, I don't know. But sometimes you fall asleep because the material is just so boring, even though it could be really interesting. Or there are other reasons, certain things that happen during the talk, or the way the material is presented. Maybe the speaker is actually really enthusiastic, really excited, but it is still boring, or it still doesn't come across, the kind of enthusiasm that the speaker has. It's not contagious. So let's look at a few problems that we might encounter with scientific talks. So one thing that you often find, especially in beginners, so people at your stage, if you're an early career researcher, is this idea that a talk should be structured like a scientific paper. An introduction, methods and materials, results, and a discussion. That works very well for a written paper, but it doesn't work very well for a talk. But you see this a lot. And usually, people will be turned off by that. What you also often find is that the speaker is not relatable and engaging. This can happen for different kinds of reasons. Maybe the speaker is someone you really don't like. That happens, right? But then maybe you pay extra attention because you hope that they make a mistake, or that they look stupid or something. So that can happen. A speaker may not be very relatable because they stand up front and they sound like a robot because they've memorized their talk. And this is also something that if you don't have a lot of experience, you might be tempted to memorize every single word. And what we often do if we don't practice appropriately, then we have a monotonous voice and we just sound like a machine, and within 5 minutes the entire room is asleep, right? So that's what we sometimes find. We might also find that the speaker is enthusiastic, is relatable. The talk may even be structured well, but the speaker uses a lot of jargon, a lot of lab language, a lot of abbreviations and acronyms, and you just don't have any idea what they're talking about. If you've ever gone to a molecular biology or biochemistry seminar, it happens all the time. You know, they show you one Western blot after the other, and after the third Western blot, it's just like this, right? So I don't go to talks like that. I'm just not interested. I don't learn anything. And quite often, and this is a dangerous thing, and I certainly fall victim to that as well, that the audiovisual aids that for us as scientists are so important, like showing graphics, showing cartoons and so on that are supposed to explain things, they can often detract from the important points because there's just too much information in there, right? So we have to keep that in mind as well. So I'm going to try to touch on these different things and how we can avoid them when we give talks. So the first thing that we need to realize is that when we get up and we talk about our research, or we summarize somebody else's research as part of a class project, is that we really want to tell a story, right? So when you were a child, chances are that somebody in your family told you stories, right? Children's stories and so on. Chances are that some of you are actually really good storytellers, right? You meet with your friends, you meet with your family, and you tell them stories about things that you've experienced, things that have happened, right? You probably don't structure that in a way where you say, here's the introduction to my story, here's the method section of my story, here are the results of my story, and here is what it means. If you tell your story like this, your family does not listen. Your friends don't listen, right? So see yourself as a storyteller. The other thing, and we'll come back to this in a moment, is you want to look at yourself as a performer, almost like an actor, right? And this is a really good perspective to take. And I don't want you to be fake. I don't want you to play somebody who you are not. But it's a good perspective to take that in a way when you are giving a talk or when you're lecturing, that you're actually putting on a performance. Because this helps particularly people who may be, based on their natural personality or so on, may be more introverted. People who find it difficult to get up in front of a crowd of people and just talk. It's difficult, right? It's very challenging. You're nervous when you do this, and if you're not nervous, then you're probably doing something wrong. Because it tells me that if you're not nervous before you give a talk, then you're not taking it seriously, right? And I'm still nervous every single time. I've done this many, many times. I'm still nervous every time. And I know that I have to, take on a particular persona to try to be engaging and relatable. It doesn't always work out. Sometimes I'm too tired. Sometimes, it's just not the right time of day, whatever. But at least that's what we should aim for. So now, if it's true that it's a performance of sorts, and if it's true that we're telling a story, then there are actually tools available, even for people who are very shy, people who are introverted, people who may struggle with the language that they are supposed to speak because it's not their native language, can get better at it. So, for example, you could take an acting class, right? Or what it really is also a useful thing to do is an improv comedy course, right? It has nothing to do with science, but it gives you tools to talk in an engaging way, to tell stories in an engaging way, and to modulate your voice in a way that will keep people engaged and focused on you and not on something crazy happening out there, or whatever the person next to you is doing. Okay, so how can we tell a compelling story? So I get invited to give talks in different kinds of departments, right? I might go to a neuroscience department, I might go to a psychology department, or I might go to an EB, Ecology, Evolution, and Behavior department. The people who do research in these different departments have very different languages, very different approaches, and very different interests. And so I need to keep my audience in mind when I put my talk together. And this is already clear from the title of the talk. So for example, people in the neuroscience department will be interested in more reductionist studies of the brain. They like to take things apart and drill down one particular problem. People in the psychology, and so a title of my talk might be Neuroendocrine Mechanisms of Aggressive Behavior, right? This is a title I've actually used talking about my research. In the psychology department, people are interested more in cognition, human and non-human behavior, right? They're not nearly as reductionist. So I might entitle my talk Social Decision Making, Genes, Brains and Cognition. In an EEB department, people are interested in evolution and animal behavior in a more organismal and integrative sense, right? They're much less reductionist in their research. And so I might call the talk Evolution of Brain and Behavior, Variations on an Ancient Theme. As it turns out, I might actually talk about the same things in all three talks. But the way I do, the way I present it, where I put the emphasis, where I go deep, or where I don't go deep, may differ depending on the audience. So the first important thing to keep in mind is when you tell a story, and you all do this naturally, keep your audience in mind. If you tell the same story to your friends or to your parents, I can guarantee you it will sound different. If it doesn't, you have a problem, right? Because you're going to use language with your parents that you don't use with your friends and vice versa. So it's the same here. Okay, and so what it means is that we want to be intentional. We want to really carefully think about our audience, and we want to figure out what is it actually that we want to get across. What do we want our audience to take from the talk that we present? So let's be intentional about it. So in the neuroscience department, a talk might look like this. This is the classic hourglass model of a presentation, where we start relatively broad, where we give background and ask the big question, but we focus in really quickly. Sometimes if you go to a neuroscience department talk, this may be the first slide, where they actually show you an animal, and that's it. And then they're somewhere down there, and very much focused on some detail. They pose a hypothesis. They go through methods and results, usually in a mixed manner for different kinds of experiments. And then they have conclusions where they come back out again, right? And if you don't know a lot about the detailed work of this particular person, you might pay attention only here at the beginning and only here at the end. And in the middle, you literally can just pass out because you're not going to miss anything. It's probably too detailed, right? But for people who work on this field, that's what they want to hear. In the psychology department, the hourglass looks a little different. We probably spend a little more time on background and big question and come up with a larger hypothesis. We go through our different results and explain how we obtain them, what methods we use. And then we present conclusions. And again, we broaden this a little bit more, we make it more relevant to a broader group of people. And in an EEB department, it may look like this. If you've gone to a lecture or a seminar on a topic of evolutionary biology or ecology or behavior, they will often give a long background. It may take 1/3 of the talk. Because people work on so many diverse things that, excuse me, we cannot make an assumption that they are on the same page. We actually have to bring them in, right? But otherwise, we have the same parts here. So that is the hourglass model. That's a useful conceptualization for thinking about your talk as you prepare it. Okay, so let's look at a simple example. I'm going to give you an example now, a very simple example. And I'll give you one version, and then I'll give you another version, okay? I'll give you a mini talk. Here's the talk. The title of my talk is Ketogenic Diet as a Supporting Treatment Option in Glioblastoma Multiforme. On a ketogenic diet, the liver produces ketone bodies, in particular beta-hydroxybutyrate, which we now call BHB. BHB can cross the blood-brain barrier and serve as an alternative fuel for brain cells replacing glucose. A diet is ketogenic when carbohydrate intake falls below 20 to 50 grams per day, while fat comprises about 80% of caloric intake. Glioblastoma multiforme, GBM, needs glucose for growth and seems inhibited by the presence of BHB. Several clinical trials are currently ongoing to determine whether ketogenic diets can slow or inhibit the growth of GMB. Do you remember anything I just said? Did you get excited about this topic? No. I mean, you just want to get out of here, right? It may actually be interesting, but you wouldn't know, right? You wouldn't even know if it's interesting. Okay, so chances are you're not interested in this. I'm disappointed, and we just talk past each other. Okay, so here's another version. Exactly the same, but here's another way to do it. And I'm doing it in the most boring possible way, but let's see how that works. The title of my talk is Why Eating Eggs and Bacon May Cure Brain Cancer. Okay? So now you have to listen because you don't want to get brain cancer. If you have brain cancer, you want to cure it. And eggs and bacon sound amazing. I mean, is it that easy? Okay, let's see. Cancer is the leading cause of death in developed countries. Despite medical advances, many cancer treatments are gruesome, yet only moderately successful. So we have a problem. If we could prevent or treat cancer by a simple change in diet, that would be awesome. Increasing evidence links cancer to high carbohydrate diets, pasta, bread, sugar, in particular sugar and starches. Eating a high-fat, low-carb diet may starve tumors of necessary glucose. Reasonable hypothesis. Initial trials with brain cancer patients have been promising, and further trials are ongoing. How was that? Is that more interesting? Yeah. If you go home tonight and you talk to your friends or to your family, do you think you can have a take-home message? Right? Give me bacon, give me eggs. Okay? I don't want your pasta. I don't want your bread. Okay? No chocolate for me, thank you. Okay? So, and I didn't even do a good job presenting the story. I didn't show you any graphics or anything. So what did I do? What I did is, and this is another way to conceptualize a story, a talk, I used the onion model. Okay? So first I told you what our problem is. Cancer is everywhere, and we're not very good at treating it. And the treatments we have are not that successful, and they're painful, right? So it's the why, the big picture. Why are we doing this, and why does it matter? You have to tell me this when you start out, otherwise I have no idea why I should even listen to you, why I should care. Okay? So that's the outer layer of the onion. We're going to peel it away and go to the next layer. That is what? We have to provide some relevant background facts and what we've actually done in our research, right? We do that for a number of reasons. First, to get us all on the same page in terms of what is known and what has been done, but also to convince you that I actually know what I'm talking about, that I'm not full of it, okay? And then finally, the details of how exactly the work was done, what kind of trial was done and what was found and so on. So that's the onion model. So let's look at the example I gave you and just basically peel away the layers. Right? So I started out with what? Spent a little bit of time of how, and then had more what. Okay? So that was in my particular example. You may do this different depending on what you talk about, depending on what your emphasis is. So that was that first example. In the second example, so here, remember in my first example, the first time I went through it, I never told you why. why you should even care about it, why you should be interested in it. So I did here in the second one, the second time I went through it, I gave you a lot of whys. I tried to really get across why we need to care about this. Cancer is terrible. We're not very good at curing it, and the cures that we have, they suck. So is there a better way to do it, right? So I talked about the what, I talked about the how. So the general advice is, and again, this is general advice, it depends on what you talk about, how much time you have, and so on, that you spend about a third or so of your talk on the why, that you spend about a third of your talk on the what, and maybe another third or less on the how, okay? So, and we can actually map the onion model onto the hourglass model like this. And somehow the colors don't quite fit. I don't know what went wrong. So that brings these two models together, and each model has a different role, right? The hourglass model gives us some idea of how we want to structure the talk overall, and the onion model gives us an idea of the kinds of emphasis we want to put on the why, the what, and the how. Okay. Now we want to dive in a little bit more and think about, most of our presentations use slides, in PowerPoint or Google Slides or whatever we're going to use for that. Doesn't really matter, and I don't care. But we have, especially in the sciences, we use visuals a lot and so on. And so far, I have not really done this, right? I've actually done everything wrong, based on what I will tell you in the next few minutes, because I gave you a lot of text, right? And we want to really avoid that. So let's think a bit about how we can build effective slides. And I give you a few suggestions that have worked well for me and for others over the years, right? This is not an exhaustive list, and some of those you may not agree with, or they may not work for you, that's okay. So my first suggestion is to basically avoid text like the plague. I have text on the slide, right? So avoid text on your slides if you can help this. Sometimes you need text. You just cannot avoid it. You just want to minimize it as much as you can. If text is necessary, all text on slides needs to be read aloud. Why is that? If you don't read the text that is on the slide, but you say something, and something different is written on the slide, your audience is going to be confused. Because some people will read and not listen, some people will listen and not read. A lot of people will try to do both, and we are not very good at that. We're actually not very good at that, right? So, if text is necessary, you should read the text aloud. I'll give you an example. The numbers here are a little bit outdated. I think they're from 2016 or whatever, but that doesn't matter. So this is my department at UT Austin in numbers. The Department of Integrative Biology has 36 faculty members. The annual grant funding is about $5 million, and the funding agencies are NSF, NIH, ARO, ONR, DARPA, DTRA, and USDA. This doesn't tell you anything here in Hungary, but an American audience would make sense of that. IB faculty members supervise 112 graduate students, 69 in EEB, Ecology, Evolution, and Behavior, 23 in Plant Biology, 10 in CMB, Cell and Molecular Biology, and 10 from other programs. 20% of our graduate students receive external fellowships, 14 are NSF GRFP fellowships, 1 NIH F31, and 10 others. So you have a pretty good idea in terms of numbers, how successful that department is compared to other departments at other universities in EEB or other departments on other topics at the same university. Okay, so that's pretty good. So realize I gave you a bulleted list of text, even though I just said we want to minimize text, right? Sometimes that doesn't help. But you also realize that I didn't throw all the text on the slide at once. I used animation. But be careful with animations. Don't have things flying from left and right, which you sometimes see, sometimes with little bing bong bong sounds. I have a colleague from UC Davis who shall remain unnamed, who does this in a way that just drives me crazy. I cannot go to a talk and listen to him. He drives me crazy. Because he has all these animations, and then he has all these many colors, and I don't even know what he's talking about. It's like a seizure. I cannot pay attention. Okay, so here's another potential way to talk about my department in numbers. And see how that is different. 36 faculty members are in my department. We generate about $5 million in grant funding from different funding sources, funding agencies. We have 112 graduate students, mostly in EEB and plant biology and a few other graduate programs. And 20% of those students are on external fellowships. See what I've done? Provide exactly the same information, but it's a lot more interesting, a lot more memorable, right? It's actually easy. Most of the time, when you have a bulleted list, you probably can do this. Not always. So, don't kill yourself. If it doesn't work, it doesn't work. But most of the time you can do it, and you can always animate it like this, right? If you don't animate it, people will read ahead, and they won't know what you're talking about. And you always want them to pay attention to you. So here's another example. Here, we want to compare bulk RNA-seq and single-cell RNA-seq. You may not know what this is. It's okay, it doesn't matter, right? But this is something that we do in my lab, and we want to compare it. So bulk RNA-seq, in bulk RNA-seq, we dissect the tissue of interest, we isolate the RNA and construct a library for sequencing, we obtain a cumulative transcript count or gene count for each gene from all cells in the sample. But the problem is that we lose the spatial resolution, and the heterogeneity of the different cells and cell types is lost. That's bulk RNA-seq. Single-cell RNA-seq, we dissect the tissue of interest and dissociate the cells, all the individual cells. We tag each RNA molecule within a cell with a unique sequence specific to that cell. We isolate then the RNA and construct a library for sequencing. We obtain specific transcript counts for each cell, which then in turn will reveal expression profiles of specific cells and cell types. So we maintain the cellular identity, and we can look at transcriptomes in individual cells. This will be quite typical of a class lecture, right? By the time the instructor is through with his slide, I'm sound asleep. Right? So how can we improve on this? Don't use text at all, but use diagrams. And again, this is usually how we do it. Most of us do that automatically, but I think it's important to really emphasize this. So here's another way. I didn't make this diagram, but I think it works better. So, I can go through the same things now with this, and I can put this on there and show you that. There's still a problem though. And the problem is that I throw all of this on there at once. And even though I may use my pointer and go through each single step, what is this? What is happening here? Right? And again, we use the same trick as before. Show your data, but don't take a data dump on your audience. Instead, especially when it's about complex concepts, complex data, build one after the other, okay? So we build many-faceted graphics. So here's the example again, and we do that. And it's not difficult to do. And you may have only one graphic that you get from a textbook or from a paper, for example, or that you made some time ago, but you can always crop it in PowerPoint or any other program. You can duplicate it and crop it, and then put it together. And so then we have a piece of tissue, and for bulk analysis, we just take the tissue and put it in a tube. We make a library and measure average gene expression from all cells, but we lose the information about cellular heterogeneity. In single-cell RNA-seq, we dissociate the cells, and each cell individually is input for the sequencing. We tag those cells with specific tags and measure distinct expression profiles, which then allows us to reveal the heterogeneity and subpopulation structure expression of thousands of cells. Right? This way we build it up, because it's complex, it's complicated. And I cannot assume that everybody in the audience is familiar with this approach. And I cannot assume that you will always just follow me as opposed to just looking ahead or looking left and right, because that's what we do, right? I do the same thing. Okay, so what else? There's a basic idea of how a slide should be structured, especially when you show data or results, right? You want to first make a statement, an assertion, and then you want to provide evidence in support of that assertion. Right? So the title of the slide makes an assertion. So slide title should not just be, here's my data. You see that quite often. Or, I can't even think of something that sounds really stupid. So instead we want to make an assertion at the slide title, and then the slide body will provide the evidence to support the assertion. I'm going to give you a very simple example of that. Here. M&Ms come in many fun colors. That is the assertion. And then I show you a picture, and that is in support of that assertion. You can now agree with me or not. You can say this guy is full of it. It is not true. The assertion is not true. M&Ms, I do not believe that M&Ms come in many fun colors. But that's a different problem. You may not agree with my assertion or the support I provide. I hope you agree in this particular case, right? But then we actually can have a conversation about it. But I first have to make an assertion, and then I have to present the support I have, or the data that I have in support of that assertion. Okay? Very simple idea, but often neglected. Another thing that you often see in talks is that people will have all kinds of different colors on their slides, right? And it can be really, really confusing to the point where you just can no longer pay attention. So choose consistent slide background. And my suggestion, not everybody agrees with that, is either all your slides should be white or all your slides should be black. Not green, not turquoise, not pink, right? I like to keep it simple. If you prefer pink, go with pink. Try it out. All I'm saying is my preference is white or black. So the advantages of a white background are as follows. It's easier to work with, especially for figures, because your figures are probably created for a white background, for a paper or a progress report and so on. So you don't have to recreate your figures from scratch, which you have to do if you use a black background, right? It's easier. So I used to use black background a lot, but as I get older, I get lazier, and so now I use only white background. Also, the slides look okay, even if the projector is weak, with a white background. And that's something that you don't want to underestimate, because quite often you go somewhere to give a talk, at a conference or at another institution, and it turns out that audiovisual equipment is just not very good. It's a tiny little projector in a gigantic lecture hall. It's all happened. With a white background, it's probably still okay. With other backgrounds, probably won't work. What about a black background? The advantage of a black or dark background is it looks more elegant. Some people don't agree with me. They don't think it's true, but I think it looks more, it's kind of cool. It's easier on the eye because you're already sitting in a dark room usually. And a dark background is particularly good with photos and micrographs. So it's actually too bright in here, as it turns out. But this is a micrograph from our lab, from immunohistochemistry, where we show you don't see anything here because there's too much light, but it doesn't matter. This is DAPI, this is tyrosine hydroxylase immunohistochemistry, and then we merge it. Against white background, you wouldn't see anything. You don't see much here because it's just too bright in the room. But with a white background, you wouldn't see anything, even in a dark room. Okay. So for photos and micrographs, a dark background is good. And so this is my one exception, because normally I will say stick to one style, white or black, to avoid visually shocking your audience. Because if you go back and forth, it's whoa, totally changed. And but the one exception to that rule is that if you usually use white background and you have a couple of slides that show micrographs or photos, it's okay to switch to black. If you prefer black background, then you just stay with black background throughout, right? Okay. Here's another thing that people often don't do, and I'm really a stickler for this. For every student, every postdoc in my lab, they have to do this. And everybody who takes a class with me has to do this. And that is to add slide numbers to make it easier for your audience to provide feedback. You may have noticed that there's always a slide number. Every single one of my slides has a number. Why is that so useful? Well, in a class context, it's obvious. Because your classmates or your instructor may actually have a question about a particular slide, and they can just make a quick note and say, hey, slide 31, I need to talk more about it. And otherwise they have to describe it, and chances are that you don't understand what they're talking about. Right? So it makes life a lot easier, not just for your audience, but also for you. And it doesn't cost you anything to add slide numbers. It's actually really true. I've noticed this myself. If you have slide numbers, you will get more questions, you will get more discussion. It may not be conscious, but that's what people do. Okay, how are we doing so far? Do these suggestions make sense? Right? They're all very simple. And yet, you rarely will see them implemented because people are not very intentional about how they give talks. That's just not something we train or we teach our students or teach ourselves. Okay? So, you may have noticed that occasionally I just show a black slide. I use black slides as a visual pause to review what has been presented so far. Right? If I turn the screen black, you have no excuse. You have to listen to me. There's no distraction, right? I mean, distractions can still happen, and I have plenty of stories of the kinds of things that can happen during a seminar or a lecture. Some are hilarious, some are tragic, but it's not the normal thing, right? So black slides are really good to pause, to review what you just talked about, to allow people to digest what has happened, and maybe solicit questions, especially if you see a lot of confused faces, right? And you don't have to stop there. You can just continue if it feels like it's not necessary. But it gives you that opportunity. Okay? So now we want to transition to something else. We talked about how we tell a story. We've talked about how we build slides effectively. Very simple rules. And now I want to give you some general guidelines. And my first general guideline will probably elicit some resistance in this audience, and I can already see it happening here, right? Because it's kind of a basic rule of a lot of scientific talks that you will start out with an overview or an outline of your talk. And I'm telling you, no, don't do this. Introduction, background, methods, results, conclusions. Avoid this. Why? Well, when you talk to your friends and you tell them about this thing that you experienced today, this funny story, do you give them an outline first? You don't. Because if you tell me a story and you first give me an outline, I'm already out the door. Because I don't have that kind of time. Right? You want to draw me in immediately, you want to get me excited about the exciting thing that has happened to you. So we want to avoid this. But I give you a break. I concede one thing, and that is, instead of an outline, consider using icons to keep your audience oriented. And you may have noticed that I have little icons here in the corner. Right? That can work. This could just be a colored square, 3 different colors or however many parts your talk has. Just to give them, oh, we're talking about something different now. Or we're going back to this other thing, right? That can give them some structure without you having to give away your entire story at the beginning. Okay? So that seems to work quite well. I don't always do that, but I'm doing it here, for the sake of making an example. And so those are the 3 icons I've used, for how we tell a story, how we make effective slides, and then some general guidelines. And I just copied these things from, after Google search or something. There's nothing magic here. Okay, what other general guidelines do we have? Remember, you perform to tell a story, but you want to be authentic. You want to be yourself. You don't want to be somebody else, right? If you think that David Attenborough is amazing on BBC talking about animals, well, that's great, but you are not David Attenborough. If I tried to talk like him, I would sound like an idiot. Right? It's just not going to work. I have to be myself. I want to tell a story, I want to be engaging, I want to be relatable, but I also want to be authentic. And it takes a while to kind of calibrate this and really figure this out. Right? You cannot be good at this from the get-go. It's just like learning how to ride a bicycle. It takes some time. You want to be relatable and engaging. And one way to draw your audience in is at the beginning of your talk to start with something like a funny little story, a personal anecdote, or an interesting fact to it. That makes it easier. Not everybody does that. You don't always have to do it. But especially if you're very nervous, or if you're talking to an audience of people that you don't know, that can really make a difference. And you can memorize this. It's only 1 or 2 sentences at the beginning that you memorize, and then you just go from there. And by the time you have told them about this little factoid, this little story about yourself, you've drawn them in, you've made them interested, you have them engaged. So memorize your opening and closing sentences, right? I think that is really helpful to get through it and to overcome your initial nervousness and to close strong, to finish strong, instead of just, you sometimes see that, and nobody knows, is he done? Is it over? Or is he about to sneeze? What's going on? Right? So, put an end to it. You cannot just fade out like a pop song on radio. You want to face your audience, not the screen. For the most part, I've tried for this entire time we've been together to look at you, right? And occasionally I look at the screen. And what I do is, this is my personal trick is, I look at different faces, and within the first few minutes, I find those in the audience who give me encouragement back. I love people who nod. Nodders are great, right? Or people who smile. Somebody who looks mean, I'm less likely to look at. I may look at you once, but I'm not going to look at you again, right? Because I want encouragement, I want positive feedback. So I know I'm not, totally off. And it keeps you going. It makes you excited about your own things that you're talking about. And this I have to qualify. So the general advice is to avoid making distracting sounds. And so the question is, what is a distracting sound? We all have these little tics. We make faces and so on. And the general advice is to avoid this and cut it out, which is actually very difficult. So when I was a postdoc at Stanford University, and I was getting ready to give job talks when I applied for faculty positions, my advisor, Russ Fernald, introduced me to this woman who was a professional in public speaking. She was the scariest person I had ever met. And she destroyed me. I had prepared this wonderful talk about my research and about my future research to introduce myself to a department that is considering hiring me. And I was very happy with the talk. I thought it was great. And she said, she's not a biologist, she doesn't know anything about what I do. In fact, she couldn't care less. And she said, give your talk. She gave me 2 hours of her time. She said, give me your talk. And she recorded it. And so I was nervous, all that. Anyways, my 45-minute presentation was over after 35 minutes because I talked way too fast. And then she just would tear into me, she destroyed me. I mean, I did everything wrong. It was just horrible, right? I wasn't prepared for that. If my advisor would have prepared me for that, it would have been better, but he did this on purpose. Right? He said, oh, this cocky guy, we're gonna take him down a notch because you certainly don't want to come across as arrogant in a job talk. And so she gave me a lot of really good advice, but you know what she did? What was pure torture in a way? She made me watch my own talk. Oh my God, what a strange voice. All these weird things that I do. What the hell? Who is this person? I could never listen to this guy. It's terrible, right? And so, but that's exactly what I suggest you do. And I do this with my lab usually once a year, where we do 5-minute presentations, so very short, we record them, and immediately afterwards, we critique them as a group. So everybody gets dissected. And they hate it. They hate it so much. And I do it too. And so, so what I've learned is a few things. One is, once you see all the weird little things that you do, you can actually begin to control them. It's not easy, but you can begin to control them. The other thing I've learned is that you start sliding again. If I have not watched myself give a talk for 2 years, I start doing all these things again that I should not be doing. Another thing that I've noticed is that there are people who may not make any strange noises. They may not have any weird tics, and it's actually more difficult to follow them. And there's pretty good research now that little pauses, filler words like or looking somewhere for a moment are actually good for the audience, so they can catch up and can process what you're telling them. So don't try to be too hard on yourself with these kinds of things. We all have these tics. It's just sometimes it can get too much, right? And I remember when I learned how to drive a car many, many years ago, I had to go to driving school. And we had practical lessons where I would drive, and we had theoretical lessons where we would sit in a classroom, just like you do. But our driving teacher was the weirdest guy. He had this weird tic where every couple seconds he would look up there. It was a tic. It was really distracting. And we all, on the first lesson, because we didn't know this, of course, we would say, what's up there? What is he looking at? What the hell is there? And of course we weren't listening to what he was saying because we were, right? Don't do that. But you may not even be aware of this until you watch yourself, right? Which is really hard. It's hard, but I think it's super important to do. And you can do this in the comfort of your home. Nobody else has to see this, right? If you're not comfortable with that. Okay, what else do we have? Don't tell jokes, but don't avoid humor. This is a really difficult one. So some people are very good at humor, and it comes naturally to them, and then you certainly should use it. But I would avoid telling jokes for a number of reasons. It can work really well, but it can be tricky. It can be very tricky. For example, you may tell a joke that is incredibly funny to you and 5 other people in the audience, but then somebody else may be really offended by it, right? And chances are you're going to have a pretty diverse audience. People come from all over the place. They have different backgrounds. It's dangerous. And you really don't want to lose them because you're telling a joke that falls flat. Again, it can work sometimes, and some of us are better at this than others, but it's probably just best to avoid it. But don't avoid humor, especially if it comes naturally to you. It can really loosen up the crowd, and it can re-engage them when things get a little slow, which they usually do at some point during a presentation. Right? Again, it takes a little practice to do that. Finally, as we say in the US, shit happens. It will happen to you sooner or later. If it hasn't happened yet, it will happen. Technical mishaps can happen anytime to anyone. They will happen to you, and it's okay. Right? It's totally possible that the projector doesn't work, and you have to do an interpretive dance or draw things on the whiteboard. It's not the end of the world. It can happen to you as it happened to me. I was giving a seminar at SUNY Stony Brook some years ago, and I was really into it, excited. People seemed interested. And then I hear this blood-curdling scream in the right back corner of the room where you're sitting, a little further back. I said, what the hell? Right? I mean, it was like this woman was getting killed or something like that. It was scary. And so what had happened was that it was, if you think your infrastructure here is not very good, if you ever complain about the infrastructure you have, it's wonderful compared to SUNY Stony Brook. Whatever it is. So here's what happened. It had rained a lot that day, and rainwater had collected in the ceiling above the room. Nobody knew. And there were these ceiling tiles, and then eventually the water was so heavy that the ceiling tiles, they got soaked with water, and one came down, and all the water rushed down on this poor woman. And there was her reaction in the middle of my talk. Now it gets better in this particular seminar. I've seen a lot, I've been around for a while. So the person next to her, people jump up and try to somehow help her, make her dry. They lead her out of the room. And as the door is about to close again, and I'm getting ready to continue my lecture, 2 stray dogs run in from the street. Nobody knew where they came from. They run up to the podium and they start barking at me, and I'm getting worried. I say, what the hell? Where are these dogs coming from? This is not normal. So it took us another 5 minutes to get these dogs out of the door. Okay, so this is just one anecdote of something that can happen. I've had people have a seizure. I've had people, all kinds of things can happen, right? And then you have to be prepared to interrupt. You have to be prepared to kind of guide, what needs to happen because you're actually in charge, or find somebody else who can take charge if you're not comfortable doing it. And then, assuming that things will calm down again, you have to find back into your storytelling, right? Things happen, it's okay. It's totally okay. Okay, so finally, you're done with your talk. Applause, you did a good job, and now it's question and answer period. So this is important. How do you navigate this period? This is a time, especially if you're a beginner in this, you haven't done this, you are scared. Oh my God, what kind of questions are going to come? What if I don't know the answer? What the hell? Okay, I would say that the question and answer period is just as important as the talk itself. remember, your audience has a different intellectual background than you do. Even if they're in your field, they have a different background. The Q&A period is a time for you to learn from them. We saw this earlier in my talk, where people ask really, really good questions, right? And this helps me not only think about my own research, but also how I might address some of these questions next time when I talk about these things already during my presentation. I can learn from them. Always repeat the question. This is particularly important now when a lot of our presentations are in hybrid mode, when people might be somewhere else on Zoom, virtually, and so on. And so you want to repeat the question. You also want to repeat the question because you want to make sure you understood it correctly. And if you repeat the question in a way that is wrong, then the questioner will probably correct you. Right? It's very easy to misunderstand a question, especially if you're nervous. If you don't know the answer, just say so. It's okay to say, I don't know. It happens to me all the time. I get all kinds of questions that I don't know the answer to. It's okay. Here's a little trick that I've learned as a university professor, as an instructor, is if I don't know the answer, you know what I do? So you ask me a question, I don't know the answer. We'll say, what do you think? Right? So I do this not because I don't want to look like an idiot because I don't know the answer. I have no problem with that, right? Because I actually don't know very much. I'm okay with that. But you ask the question, chances are that you've thought about it a little bit already, and you might actually have a good idea. And so we can enter into a dialogue in this way, right? And I might learn something, and you might learn something, and we might get other people engaged in the audience, right? Keep the tone and the scope of your answer similar to the tone and scope of your talk, right? Don't get carried away on a totally different topic. And don't, if you get a really aggressive question, which has happened to me as well on numerous occasions, don't answer the question in kind. You don't want to have, come across as arrogant or aggressive or so. Just say, well, that's an interesting question. Here's what I think about it. Basically ignoring the fact that this person may be an unpleasant person. Because chances are there is an unpleasant person in the audience. It happens, right? So it's another good thing to keep in mind. If the questioner disagrees with you or tries to hijack your talk, they interrupt you in the middle and say, well, this is more of a comment than a question. And then they go off on this long tirade, right? Or it looks like there will be an argument, then diffuse the situation. Say, well, this is really interesting, but can we talk about this later, right? Or I will come to this in a few slides, even if you don't, just because you want to stay in control. You want to stay in control. And there are people like that. Not because they're necessarily negative or overly aggressive or bad people. Some people just don't have a filter. Or they get so excited that they think they're the center of the universe and you need to listen to them. Even if whatever they want to talk about is only peripherally related to what you're talking about. So you need to learn to handle those kinds of people. By the way, you do that when you tell stories. And some of you are probably better at this than others, but you can practice this, right? You may sit with a bunch of friends and you tell a story of something that happened to you. And you probably all know this one friend who always hijacks your story. They always make it about themselves. And this is a really good, they may not be a bad person, but they have this tendency, chances are they don't even realize that they're doing this. And this is a really good opportunity to learn this, to be in charge, to stay in control, right? And say, let me finish. Or, no, it was not like that. Just listen, then you learn what actually happened, right? And yes, this is really interesting. I want to hear about this later, but let me finish my thing first. Those kinds of things. Do not insult the questioner. Say, well, that's a stupid question, right? It may be a stupid question, but that doesn't give you the right to say this. And I feel very strongly that there are no stupid questions. right? When somebody asks a stupid question, chances are that it reflects a failure on your part that you didn't explain something very fundamental. And you can learn from that. And you might feel it's a stupid question, but don't tell them that. You say, well, who are you? You know, what makes you, why are you even here? That kind of thing. But don't do that. Don't do that. Okay, so do not put down the work of others. Instead, highlight the positive aspects first before pointing out limitations and gaps. You may set up your story and say, nothing is known about this. Chances are that's a lie. Or you might say, everything that has come before me was just useless. Don't do that. You know, even if you disagree with this earlier research, even if you think there are huge gaps in our understanding, you can say this in a positive way. People have thought about this. People have put some effort into it. However, there's still a lot to be learned. And then it's a lot less adversarial, right? And similarly, and this is really a corollary of this guideline, is don't oversell your own research. Keep it honest, okay? And now, when you finish when you're done, and preferably sooner. You know, don't do what I'm doing, don't go over time. It's Balash's fault. Finally, practice, practice, practice. Not just this one presentation that you practice in front of a mirror or with a willing friend or something like that, or your lab group. Practice, practice, practice also means every time you get an opportunity to give a presentation, you take that opportunity and you do it. You get better at it. It's really like learning how to ride a bike. All right. So, here are the take-home messages. Tell a compelling story, build effective slides, be authentic, be prepared, and be honest. Okay? And that's it. I got some ideas from David Stern, some ideas from Klaus Wulke at these websites. This is my lab group. They had to listen to this a couple times and gave me a lot of feedback, so I thank them for that. And I thank you for your attention. Let me know if you have any questions. Thank you.
Any questions? Is it a good sign that you don't have any questions after the talk?
It's after 6 in the evening.
Yeah, probably.
They want to go home and not listen to some
But in
strange
general,
guy.
so you know that sometimes you don't get any questions after the talk. What do you think of that situation?
Well, if I want to feel good about myself, I say I've explained everything so well, there are no questions. Or I say they really couldn't care less about what I have to say, you know, whatever. I'm not too worried about it. Yes.
I have a question about opening and closing sentences. What are the most, can you give us some examples what to avoid when we are trying to generate our closing sentences?
Yeah, don't make it complicated. Keep it simple. Like, just, you know, maybe what I said. You have some final message, some concluding statement about your research or whatever, or what you presented. And then you might say, thank you for your attention. I would be happy to answer some questions. And that's what I usually do because it's very simple. And I usually try to put some emphasis into it so that the people who've fallen asleep wake up again. Yeah, because people do fall asleep. Yeah.
Professor, I would like to ask, when you prepare a slide for research, sometimes you have a methodology and you have so many steps, and then you have the main big result at the end. And I find it like logical to use to talk about the result, the methods and the results at the same time because it's easier to explain.
Mm-hmm.
However, in this situation, what do you suggest? Should I each step of methodology and talk about what we found, or just talk in general what we've done and speak of the big results at the end? So
it depends a little bit. If the focus is really on this one experiment, this one result, and the audience, like in a class or in your lab, your supervisor, they really want you to lay out the experimental steps in some detail, then you probably will have to do that before you get to the result. But you can again do this, maybe see if you can do it graphically, right? In the form of a flow diagram, for example, that has some graphics in it that make it more interesting. You can do this in an animated fashion, all right? It doesn't have to be like a written protocol. You can turn this into something more interesting. If you give a bigger talk where this is only one part of a larger story, then you may just be very abbreviated and say, I did quantitative real-time PCR, and if you have questions about the method, we can talk later, and just move into the results. Or just give a basic cartoon of how qPCR works, very simplified, and move in. It depends a little bit on the context. Again, for a class, this may be a little different because there may be a clear rubric on how you have to do it. And then you should follow that rubric. But often instructors fail to communicate what their expectations are, and then they give you a hard time about it. They say, well, you didn't tell me, right? So, and then you are totally, as a student, you're totally within your rights to say, hey, you did not give us a rubric on how we are supposed to do that, right? We can do this in a friendly way so they're not offended.
Well, honestly, Professor, sometimes 10 minutes presentations.
Yeah. They are really difficult. These short presentations are hard. They are among the most difficult ones. And you don't want to spend much time on methods there at all. You want to give a really nice, setting up, big question, background, why this is interesting, why this is important, what is your hypothesis or your prediction, and then go into show your results, how you do the experiment, experimental design, what your results are, and you say what the methods are that you've used, but you don't do any more than that. And then you zoom out again and say, this is what this means. How I think about it for the larger field or whatever. Does that make sense?
It does, actually. Thank you so much.
Good. Anything else? Yeah.
So my question is, how should we initiate the presentation? Like, what will be the effective attention gainer so we have the audience and we can continue?
Well, it could be something very simple. So, for example, you could say, I'm the one who stands between you and your lunch, right? And which may happen because you're the last speaker in the morning session. Or in the afternoon, you might say, I know you'd rather take a nap now, but I appreciate you being here and listening to me. Right? So that puts everybody at ease. It gives them some permission to kind of nod off if they really feel like it. But it also moves you over this hump of the initial nervousness. Right? You can come up with better ways. Everybody can. You don't have to use what I'm telling you. But it's useful to think about it ahead of time. And to kind of say, think about it. What is it that I'm going to say? Or you might say something like, okay, you've heard all about exciting topic A, B, and C in the preceding talks. I'm going to tell you about something completely different. And if I'm in the audience, I say, whoa, that should be interesting, something completely different. I have no idea what it is, but it should be interesting, right? So those kinds of little tricks, yeah.
Thank you.
And don't overthink it. Keep it simple, right? Yeah.
I would have a question that what we could do if we freeze in a presentation because we overstress the situation.
If you freeze in the presentation,
yes.
Yeah.
And I don't know what to say in the next moment. What can we do to get back to the way?
Yeah, I've seen this happen. I've seen this happen to really, experienced people that they look at the next slide and they just have no idea what they are supposed to say. I don't know if I have any good advice for this, other than to just wait for that moment to pass. I'm not sure what else to say. It does happen, but I'm not sure. Maybe somebody else has good advice on when that happens.
Inhale, exhale.
Pardon me?
Inhale, exhale.
Yeah, that's good. Breathe. That's actually really good advice. Yeah. Just breathe and try to stay calm. And chances is just a temporary kind of paralysis of your nervous system. But again, if you structure your talk carefully, if you really think about your audience, if you follow some of this advice, I think these kinds of situations are much less likely to happen, right? Because the big difference is that, and again, you don't have to follow all this advice, to the letter, and some things you may not agree with, but some of the things you probably will find useful. And so the trick is that you want to stay in control of yourself, in control of your talk, in control of the audience. And I think the freezing is a sign that you're losing that control. Right? It can happen. I mean, and if it happens to you, it happens to you. It's okay. Nobody will give you a hard time, or if they do, then it's their problem, not your problem. And tomorrow they will all have forgotten about it. You feel terrible about it, but it's okay. It's okay. I've had the most embarrassing things happen in my life, and I don't look back. It just happened. Shit happens. Okay. Anything else? That's it. Well, I really appreciate you turning out this late in the day, and I hope you all have a good evening. Thank you.
Yeah, thanks again.